Very good to see you all. Thank you for coming along to this event. As Will said, for us, the decision to organise this workshop grew out of discussions Olwen, Will, Richard, and myself were having about interdisciplinary projects. And one thing we realised fairly early on is that it's important to get a common understanding of the problem which is being discussed in these sort of discussions before looking at more creative ways of tackling them. So really, as a preamble to the more specialist talks in this meeting, we thought it'd be a good idea to just start by running through the biology of the problem here. So the basic concepts to which we hope we'll be able to find biophysical solutions. Now, obviously, we appreciate this is by design an expert workshop, and that means that all of you will be very familiar with some of what's going to happen in the presentation I'm about to give, and some of you, I'm sure, will be familiar with all of it. But it's a helpful start to all these discussions to make sure everyone has a common set of concepts to work from. So that's the purpose of these slides. And the first thing I'd like to emphasize is the scale of the problem we're facing here. So influenza, as you're all well aware, is an enormously important global pathogen. Its tendency to cause pandemics probably attracts the most concern and the most attention, but even as a seasonal disease, it has a massive impact on health. These are data here from the United States, which is both a developed country and one in a temperate region, in which you can see a fairly typical pattern for countries of this sort of a significant burden of severe illness, including deaths, impacting particularly older adults, but also at-risk groups such as the very young, the immunocompromised and the pregnant. It's worth pointing out as well that those severe cases are really the tip of an iceberg of morbidity, which impacts on society in many ways and has enormous economic cost. Although we tend to focus on developed countries in temperate regions, it's worth bearing in mind that actually globally this is not a fully representative picture of influenza, and that probably the highest burdens of influenza disease fall in the tropics in particular, where for a variety of reasons the impact of disease is higher, and where influenza has a more complex distribution throughout the year. It's also worth highlighting, of course, influenza is not merely a disease of humans, it's a very widespread disease among many vertebrate hosts, and whilst most of the human disease is due to influenza A and B viruses, important domesticated animals are infected by A, B, and D types of influenza as well. And this is also a major economic cost for us. Now, this is not a new problem. Influenza and severe disease caused by influenza has been an ongoing feature, well, probably since the domestication of animals, and certainly from the age of modern medicine onwards. And as a result, influenza has attracted close study for a very long time. Avian influenza was one of the first animal pathogens to be identified as being caused by a virus. Human influenza was one of the first human pathogens to be identified as being caused by a virus some decades later. It proved to be very easy to culture, which means we've been able to study it for some time. We've had over 20 years of robust reverse genetic systems, which have allowed us to precisely genetically engineer the virus. And that application of technologies and that focused interest has paid off. We've had vaccines in many respects similar to some of those used today. Since the 1930s, we've had, by virus standards at least, a number of licensed drugs for treating the more severe cases. But despite this, and for reasons we'll get onto, this remains an ongoing global threat. We also now understand in considerable detail what is causing this disease, the virus itself, the components that we understand in essentially molecular detail now. And in terms of vaccination, we'll be focusing this talk particularly on these spikes on the surface, the hemagglutinin HA and neuraminidase NA. It's worth flagging up, because this is one of the points which is put in for discussion, that these are rather idealized virus particles, although ones shaped like this have attracted the most focused attention in laboratory. Influenza and clinical infections can cause a huge range of virus shapes to be produced, including these beautiful, if bewildering, filamentous forms whose importance in infection remains, I think it's fair to say, fairly obscure. So if we run through then what the building blocks of the virus are and where the opportunities are for understanding the biophysics of these. So in humans, at least, influenza is spread primarily through the respiratory route. So an infected person will be breathing out both aerosols and droplets containing virus particles, which can either directly infect people or which can contaminate fomites and then lead to infection through that indirect route. The importance of this for influenza transmission can really be highlighted by looking back over the last 18 months, where the sort of non-pharmaceutical interventions required to mitigate the effects of coronavirus have caused influenza incidence to crash to fractions of their normal levels. Once inside a host, the influenza virus particle enters the upper respiratory tract, enters mucus, and from there it needs to find a cell to infect. It will do so by binding to it with the hemagglutinin spikes on its surface, which will bind to the viral receptor. It's classically thought of as sialic acid, although as Rob DeVries will talk about soon, that's perhaps a slightly simplified picture, but sugar modifications to proteins and lipids. Now, these are actually also present in large amounts in the mucins, in proteins present in the mucus. So influenza at first has to diffuse around interacting with those for a series of transient interactions until it reaches the cell surface and can move from diffusing in 3 dimensions to moving in 2 dimensions across the cell surface, which it does in something of a random walk, until it reaches a point on the cell surface where the cell will undergo receptor-mediated endocytosis, engulf the virus and take it up into the cell interior. At this point, the compartment containing the virus undergoes changes, both kinds more acidic and has an influx of potassium ions as well. That increase in acidity causes radical conformational changes in the hemagglutinin. So we can see here the blue head domain, which is bound to the receptor, that green blob. The stem of hemagglutinin rearranges to thrust a fusion peptide into the membrane of the endosome of the cellular compartment, and then these stems fold back against each other. And when multiple hemagglutinin spikes do this simultaneously, this releases enough energy to bring the membranes together and fuse them. And this has been now studied in enormous detail. We can see an image here from a very nice recent study from a scale group showing how the fusion process works, and including now details of how that works with the transmembrane domains of this as well. There then occurs a lot of basically complex cell biology, which is unimportant, I think, for discussions we're having today, or at least which can be safely put in a black box, but which essentially means that a virus goes in, and then somewhere between 6 and 18 hours later, lots of viruses start coming out from the surface of the cell. They face an immediate problem, which is that they're being released onto the surface of the cell where previously they would have bound and entered. And so the virus needs to degrade and remove the receptors which it bound to previously to enter the cell. This is done by the other major spike glycoprotein on the surface, neuraminidase, which enzymatically cleaves off sialic acid, so these sugars which the virus binds to. And the effects of this can be seen when we look at the effects of neuraminidase inhibitors, which include some of the major licensed antivirals, such as Tamiflu or oseltamivir. You can see that causing virus particles to aggregate at the cell surface rather than be properly released into the mucus. Whilst this is happening, the body is also, of course, reacting, and there are 2 main arms for this. One is that viral proteins within the cell are chopped into bits and then presented on the cell surface where they can be recognized by T cells, which will kill the infected cells. The other is by the production of antibodies, which will bind. Well, we think this predominantly in terms of binding to the hemagglutinin and neutralizing its ability to interact with receptors on the cell surface. As this figure shows, actually, there's a lot of ways in which antibodies can interact with a virus particle in order to direct an immune response against it. But the capability of antibodies binding to the virus particle to prevent and limit infection is why so much focus has gone into developing vaccines which will effectively target those proteins. And it's a moving target to aim for, because the virus is mutating extremely rapidly, as you would expect. Those spike glycoproteins are changing constantly. And this figure here, put together by Will, shows the necessity of repeatedly updating the vaccine in order to match changing hemagglutinins and neuramindases on the surface of the virus. And this meta-analysis shows why this is important, because the effectiveness of vaccines falls off rapidly the more quickly, the further separated the virus is from the vaccine which it's trying to match to. In order to keep up with this constant change of virus, there is a worldwide monitoring effort to understand how which influenza viruses are circulating. And this feeds into WHO decisions as to which vaccine should be produced. But after that decision is made, vaccines do need to be produced. And this is a long process. There are a number of methods now there, but the classical methods, and you can see on the top, the egg-based production method, which still accounts for the majority of the global supply, will typically take around 6 months from the announcement of a strain to being able to manufacture at scale the vaccines needed to protect people. Fortunately, at least in temperate regions where at least the larger part of the vaccine market is, if not the larger part of the burden of disease, transmission of the virus between the north and the south and the fact that the virus spreads most effectively in the winter months means that what's happening in the northern hemisphere in its winter is a reasonable predictor of what's happening in the southern hemisphere in their following winter, and vice versa. And as a result, we find ourselves in this situation. This is a somewhat complicated figure, basically spanning out a typical year of influenza prediction. I'd like to draw your attention firstly to the coloured asterisks I put on here. So if we look here at pink for the southern hemisphere, we can go from a strain selection meeting and then 6 months of preparation to vaccine distribution the following winter. And more immediately afterwards, a northern hemisphere strain selection meeting. It's 6 months of frantic work in orange there before vaccine distribution begins in the north. And this process continues year on year on year. Indeed, the current VCM meeting for the northern hemisphere is underway as we speak. And sometimes this works well, and sometimes, as you can see from this graph, just tracking influenza incidence in the US year on year, it works less well. There are a number of reasons for that, some of which fall without the scope of this meeting, such as the logistics of distribution, issues relating to vaccine hesitancy, some of which are ones which can be addressed through vaccine design, so producing novel vaccination approaches which can offer longer lasting immunity and which can try to protect us against the radically new viruses which appear during pandemics. But even for the seasonal viruses which we update year on year on year, our ability to make accurate predictions of what they should be 6 months ahead of time is more limited than we would like. The vaccines save a huge number of lives, but if they were better, they could save a great deal more. And that's the challenge which we're meeting here to discuss. So currently the effectiveness is variable. There are challenges in predicting the circulating strains 6 months ahead, and in challenges in predicting how vaccines selected that far ahead will perform in practice. And the purpose of this workshop is now to take those challenges, those questions, and to discuss the capabilities of biophysical methods for improving our ability to make those predictions and make those responses. So with that, I will wrap up this initial primer and hand back to the next stage of the meeting. Thank you.